I hope you're good. I'm good myself. Without wasting any time, so today we'll be doing paper two, right? For those who are writing paper two, final paper two this June. So this question paper is forwarded, right? So this was a November 2024 question paper. Uh, so I hope it helps you, it assists you to be able to know how to approach questions. Okay, let's get into it then. You know that you have to read the instructions whenever you're about to write a question paper, right? Make sure that you read the instructions so that you don't make mistakes. Okay, let's start with the multiple choice. So our multiple choice reads as follows, right? Start with question 1.1.1. Obviously, you have to read the statement, but I won't do that here. I'll just read the questions. You already know what you're supposed to do with regards to multiple choice. Okay, let's get to the question. Which one of the following organelles is the site of protein synthesis? Well, that one is easy, right? But the first thing that you need to do if you're not sure, right, is to rule out, right, the options. So let's start with A. So A states that it's the ribosome. So we know that A is the right answer. So the site of protein synthesis is the ribosome. Okay, let's go to B, chloroplast. So chloroplast, we do not have, humans do not have, or human cells or animal cells, they do not have chloroplast. You only find chloroplast in in a plant cell, rather. Does that make sense? Mitochondria. We know that mitochondria, protein synthesis does not take place in the mitochondria. What takes place in the mitochondria? Cellular respiration. So you did cellular respiration in grade 11. Right, okay, now let's go to D. So D, centrosome. Definitely that's not the answer. We only see centrosomes in life sciences when we get to meiosis, right? Do you still remember those structures that form centrioles? Okay, so our answer here is A. Okay, answer here is A. So let's go to 1.1.2. Let's go to 1.1.2. Okay, in humans, the gonosomes determine the gender. Remember, what are gonosomes? Gonosomes are sex chromosomes. If you still remember, they're sex chromosomes. So you have autosomes, autosomes from pair 1 to pair 22. Then pair 23, we have gonosomes. So gonosomes are sex chromosomes. They determine sex, right? But when it comes to from pair 1 to pair 22, we call those autosomes. And they deal with the functionality and the characteristics, right, of a cell. Does that make sense? Okay, so let's continue. Which combination below shows the correct gonosome for males and females? Okay, correct gonosomes. So here, A, let's go to A. Already we know the answer. You should recite the answer in your head before you even read or go to the question. Okay, let's go to A. So A, male chromosomes, XY is correct. Now let's go to female chromosomes, YY, it's incorrect. So we're ruling out A. And let's go to B. B, male chromosomes, right? YY, it's wrong, right? It's not YY. Let's go to female chromosomes, XY. It's wrong, we're ruling out B. So now let's go to C. C. So C reads as follows: XY, male chromosomes, it's correct. Then when it comes to females, XX. Yes. So our answer becomes what? C. So our answer is C, right? But let's check out D, right, just to make sure. So D, male chromosomes XX, already this option is wrong. So you cannot trust this option. So it's wrong already. Okay, let's move on. Let's continue. Okay, 1.1.3. An organism has the genotype homozygous dominant. So it is important for you to know, right, and familiarize yourself with regards to using such words, right, such terminology. So let's continue. 1.1.3. An organism has the genotype homozygous dominant. So we call this homozygous dominant, right? Homozygous dominant because the two alleles are the same. Homo means same. Okay, let's continue. The correct term which describes this organism's genotype is heterozygous dominant. I've already told you the answer. So I've already told you the answer, right? So the answer is homozygous dominant. So it's C. So there's no point in going through the options. Already I've told you the answer. So it's easy. Easy multiple choice. These are free marks, guys, right? So 2 marks each question, totaling 20 marks if there are 10 questions. Okay, let's continue. Let's go to 1.1.4. Down syndrome is a genetic disorder, right? Where an individual has an extra copy of chromosome 21, or we can call this trisomy 21. Does that make sense? So which one of the following may lead to Down syndrome? So let's look at A. A reads as follows: failure of chromosomes to replicate during mitosis. Already it's out. Mitosis, Down syndrome is not caused by mitosis. So A is out. The moment they mention mitosis, it's already out. So let's go to B. B, failure of chromosome pairs to reseparate during meiosis. Okay, this is closer to the answer, right? Failure of chromosome pairs to separate during meiosis. And we call that what? Non-disjunction, right? Okay, now let's look at other options before we choose B. C, failure of chromosomes to form pairs during fertilization. It's wrong. It's wrong. Anything that does not include meiosis, it's out of the picture. Let's go to D. Failure of chromosome pairs to cross over during meiosis. So it's wrong. So our answer is what? B. So our answer is B. Answer becomes B. Okay, let's continue. What am I doing? Let's continue. Okay, 1.1.5 reads as follows. A gradual change, the moment they speak about a gradual change, they're speaking about evolution, right? Gradual means a slow change. A gradual change in the characteristics of species over time is known as, okay, A, punctuated equilibrium. No, we know it's not punctuated equilibrium. Remember, what is punctuated equilibrium? We stay a long period of time without change, and we have that instantaneous change, right? That happens, right? Due to a mutation or whatever, but we have long periods without change, right? So the diagram will be like this. So we have long periods without change. Then we have a spark here, change, just have a punctuated equilibrium. Then long periods without change. Then we have punctuated equilibrium. So, so this is a graph of punctuated equilibrium would be like this, right? I'm just trying very hard to portray that to you, even though I'm not um, doing it to a sufficient manner, right? Okay, let's continue. So it's not punctuated equilibrium, because punctuated equilibrium you stay long periods of time without change, right? So there's no slow change. It's just long periods of time without change, and then we have that punctuation. Hence, it's called punctuated equilibrium. We have that change that happens instantaneously and randomly, right? Okay, so that's punctuated equilibrium. So let's go to B, genetic engineering. Definitely, it's not genetic engineering. Genetic engineering has to do with the modification of an organism's genes, right? In order for benefits to, with regards to agricultural use, industrial use, or medicinal use. So it has to do with the manufacturing of an organism's genes or genome, right? For industrial use, uh, agricultural use, or medicinal use. Does that make sense? Okay, let's go to C. So C states, reads as follows: speciation. Definitely it's not speciation. Speciation is the formation of a new species. So speciation is the formation of the new species. So it's not C, which leaves us with D. So our answer is biological evolution, right? Okay, let's continue. Let's go to 1.1.6. 1.1.6. Which one of the following is a reproductive isolation mechanism? Okay, reproductive isolation mechanism. Let's go to A. So A reads as follows. Adaptation to the same pollinator. Definitely, this is not a reproductive isolation mechanism, right? Because it does not isolate organisms, right? So it does not. So if it said adaptation to different pollinators, then it would have been a reproductive isolation mechanism because it separates, right? Organisms, it separates animals, right? Okay, let's go to B. Species specific courtship behavior. B is closer to the answer, but let's look at other options. C, production of fertile offspring. Definitely, that's not true, right? Actually, it's the production of an infertile offspring. Okay, let's go to D. Breeding at the same time of the year. No, it's breeding at different times of the year. So it leaves us with B. So our answer becomes what? Becomes B. Okay, let's continue. Let's continue. 1.1.7. Analysis of mitochondrial DNA is an example of the line of evidence. Okay, let's go to A, mitochondrial DNA. Let's go to A. Already we know the answer, but let's go to A. So A goes as follows. Fossil evidence. We know that's not true, right? Mitochondrial DNA has nothing to do with what? Fossils. What are fossils? These are remains of dead animals or plant matter, right? Okay. So you can find such fossils in rocks, you can find others in ice, right? So definitely mitochondrial DNA is not part of what? Fossil evidence. So let's go to B. Modification by descent. Definitely it's not. Modification by descent, right, is the same as evolution, right? As the time goes by, organisms they change. They become different from the common ancestors. They are no longer the same as the common ancestors. So that's the modification. That's modification by descent. So as you descend from the common ancestor, you become different. So modification means change. So you change from the common ancestor. So to descend is to come from something. Okay. Let's go to C. Biogeography. So, but it's not true. Biogeography is just the distribution of organisms throughout the whole world. It's the distribution of organisms throughout the whole world. So it has nothing to do with mitochondrial DNA. So the answer becomes what? D, genetic evidence, because it has to do with genetics. Okay, let's continue. Let's go to 1.1.8. 1.1.8 reads as follows. Which option in the table below shows the correct comparison between mitosis and meiosis? Okay, let's go to A. A reads as follows, produces 4 daughter cells. That's mitosis, which is wrong. Already it's wrong. We know that mitosis produces 2 daughter cells. So we're ruling out A. Okay, B produces genetically different cells. Nope, that's wrong. Mitosis does not produce genetically different cells. It actually produces genetically identical cells. So already we're no longer going to meiosis because we know with the B is already wrong by just reading that statement. Now let's go to C. The chromosome number remains the same. Okay, true. The chromosome number remains the same. If you're starting with 46, you're ending with 46. So there's no halving of the chromosome number. Then let's go to meiosis, right? The chromosome number is halved. Yes, so C is closer to the answer, but let's go to D. So D states as follows. 2 divisions occur. Nope, already D is wrong, right? Actually, 1 division in mitosis occurs, right? Okay, so our answer here is C. Okay, let's go to the next one. So 1.1.9. So which one of the scientists discovered the Darwin child, rather? Which one of the following discovered the Darwin child? So the answer, let's look at the options, right? So the first one is Lee Berger, right? So Lee Berger definitely not the answer, right? The second one is Ron Clark. Ron Clark is not the answer. The third option is Raymond Dart, right? So Raymond Dart definitely is the answer. So our answer becomes what? Becomes C. Does that make sense? I hope it does make sense. Let's go to 1.1.10. Let's go to 1.1.10. So you need to know the names of these fossils, of these scientists. What am I saying? I'm saying fossils now. Okay, of these scientists. Okay, so let's go to 1.1.10. Which one of the following is produced at the end of translation? So which one of the following is produced at the end of translation? Okay, remember, what is translation? So we have two stages in protein synthesis. The first stage is transcription, and the second stage is translation. So what's produced at the end of translation? It's the protein, right? So what's produced at the end of translation is a protein. So our answer here becomes what? Protein. So it's C. The answer becomes what? C. Okay, let's continue. Let's continue. Okay, let's go to biological terms. So biological terms reads as follows. Let's start with 1.2.1. So 1.2.1 reads as follows. A nucleic acid that carries hereditary information. That's easy. So we know that the answer there is DNA, right? DNA. So the answer here in 1.2.1 is DNA. Okay, let's go to 1.2.2. The chromosome condition of a cell that has a single set of chromosomes. So the moment they say single set, that's haploid. So that's the condition, haploid. Double set or two sets of chromosomes, that's diploid. Right? So a single set is haploid. Let me just remind you how many chromosomes is haploid. Right? It's 23 chromosomes with regards to human beings. 23. Diploid number is 46. Haploid number is 23. Okay, let's go to 1.2.3. Right? So 1.2.3 reads as follows. The structure that holds the two chromatids of a chromosome together. So that's easy. The answer there is centromere. Right? Remember, so it's actually not a structure, but it's a point at which two chromatids meet. So it's a point. Okay, let's continue. Let's go to 1.2.4. So 1.2.4 reads as follows. The type of RNA that carries specific amino acids to the sites of protein synthesis, right? I'll repeat that again. The type of RNA that carries specific amino acids. The moment you hear the word amino acid, the only RNA that carries amino acids is the tRNA or transfer RNA. And also be careful when you're writing transfer RNA, use a lowercase t. Use a lowercase t. Okay, let's continue. So 1.2.5, the analysis of DNA samples to identify individuals or relationship between individuals. Whenever they mention DNA samples, right, where they analyze DNA samples, we call that DNA what? Profiling, right? Call that DNA profiling. So that's the technique. Okay, let's continue. 1.2.6, a representation of the number and structure of all the chromosomes that occur in the nucleus of a somatic cell. So the answer there is karyotype or karyogram, right? So karyogram is a combination of karyo, which relates to the nucleus, and diagram. So you can either say karyotype or karyogram. Okay, let's go to 1.2.7. A group of similar organisms that are able to interbreed to produce a fertile offspring. So the answer there is species, right? So species, they share similar characteristics and they're able to interbreed to produce a fertile and viable offspring, right? Okay, let's go to 1.2.8. The phase of meiosis where paired chromosomes are arranged at the equator. Paired chromosomes arranged at the equator. The moment they speak about equator, it's metaphase. But you're not supposed to just say metaphase. So we have metaphase one and metaphase two. What's the difference between metaphase one and metaphase two? So metaphase one, the homologous chromosomes, they are arranged in pairs. Right? They arrange at the equator in pairs. Whereas in metaphase two, they are arranged in the equator singly. So the answer here is metaphase one because they said here where paired chromosomes are arranged at the equator. Okay, let's go to 1.2.9. 1.2.9 reads as follows. The biotechnological process that produces genetically identical organisms. So the process there is cloning, right? The process is cloning. Okay, let's go to 1.3. So guys, before I move on, just make sure that um, you study, you revise your biological terms. Just go through your biological terms, you'll be fine, right? Make sure that you go through your biological terms. They're there in your textbooks and everything. Oh, so I hope that's okay. So let's go to 1.3. 1.3.1. A source. I won't read the statement, the instructions. You already know the instructions when it comes to 1.3, right? Okay, let's go to 1.3.1. A source of variation in populations. A, random fertilization. So random fertilization is a source of variation, right? When different gametes, right, with different genetic material fertilize each other, right? Okay, so let's go to B, random mating. Yes, that's true. That's also a source of variation. So the answer here is both A and B. Please make sure that you mention that it's both A and B. So it's important for you to mention that it's both A and B. If you just say it's A and B, then it's wrong. Okay, let's go to 1.2.3. 1.2.3. Okay, 1.3.2 rather. 1.3.2. Discovery of the structure of the DNA molecule. So A, Franklin and Wilkins. B, Lamarck and Darwin. So Lamarck and Darwin, they're involved in evolution. They have nothing to do with the DNA structure, with the discovery of the structure of the DNA molecule. So the answer here is A only, Franklin and Wilkins, right? And also it's important for you to learn about the discovery of the DNA, right? And to learn about the fact that who got the Nobel Prize, right? So it's important for you to understand the discovery of the DNA molecule. Okay, let's continue. Let's go to 1.3.3. Occurs during meiosis. A, karyokinesis. B, cytokinesis. So remember, karyokinesis is the division of the nucleus. Anything that has to do with karyo indicates the nucleus. It has to do with the nucleus, right? So karyokinesis happens in meiosis, right? Let's go to B, cytokinesis. Cytokinesis is the division of the cytoplasm. It also takes place during meiosis, right? Actually, these processes, they happen simultaneously. So the answer is both A and B, right? So let's revise the answers again. 1.3.1, the answer is both A and B. 1.3.2, the answer is A only. 1.3.3, the answer is both A and B. Okay, let's continue. Now let's go to the question. Let's try to analyze the structure. Let me just zoom this out for you to see, or should I zoom it in? Okay, let's try to analyze the structure. Yeah, let's try to read the statement. The statement is very important because they tell you, they hint to you. So the statement hints, right? Before you answer the question, please analyze the statement. Okay, the diagram below represents a DNA molecule undergoing an important biological process. So what's this biological process? So we can see by the look of things here that both DNA strands are involved in this process. The moment both DNA strands are involved in this process, it becomes what? DNA replication. So what is DNA replication? DNA replication is the process whereby the DNA molecule makes an exact copy of itself. So you can see that this is the DNA replication. Why? Because the two strands are involved, right? They act as templates. So the two strands, they act as templates. So let's try to look at one. What is one here? So one becomes what? It's in between nitrogenous bases. Obviously, it becomes what? A hydrogen bond, a weak hydrogen bond, to be specific, to be precise, right? Okay, let's look at two. Two becomes what? It becomes the phosphate ion, or the phosphate group. You can say that becomes the phosphate group. Then 3, right? 3 becomes the pentose sugar, or you can say the oxyribose sugar, because we've identified that this molecule is the DNA molecule. Actually, they told us at the top there, with this molecule is the DNA molecule, right? So we know that 3 is the pentose sugar or the deoxyribose sugar, which belongs to the DNA. So 4 becomes what? Becomes our nitrogenous base, right? And then the rest of the structure, right, that includes 2, 3, and 4 is called what? A nucleotide. What are nucleotides? Nucleotides are monomers or building blocks of the DNA, right? So they are monomers or building blocks of the DNA. Let me just erase this. Okay, so let's continue. Let's go to questions, right, and see what they'll be asking us in these questions. Okay, so identify the process shown in the diagram. Already I've told you the process, that the process is DNA replication. So the process becomes DNA replication. So 1.4.1, identify the chemical bond labeled 1. So already identify the chemical bond label 1. We identified it. So the chemical bond becomes what? The weak hydrogen bond, right? The weak hydrogen bond. Okay, let's continue. Let's go to 1.4.2. So give the collective name for the parts labeled 2, 3, and 4. Okay, we've already identified that the collective name is nucleotide. So that's the collective name. So nucleotide. Okay. Let's go to 1.4.2b. Give the full name of the nitrogenous base label 5. Okay, let's check the nitrogenous base label 5. Let's see. Okay, there is it here. Labeled 5. Where is it? One eye. There is it. Okay. So the nitrogenous base label 5 becomes what? It has to correlate with, let me just count. Let me count just to make sure. Let me count. Let me just remove this. Let me count just to make sure. Okay, count. Let me count the nucleotides. 1, 2, 3, 4, 5. Okay. Let me count this side. 1, 2, 3, 4, 5. Okay. So a nitrogenous base 5 becomes what? Thymine. So it becomes what? Thymine. Let me just write the T here. Becomes thymine because it has to match with the with this strand, with this original strand, right? So it has to match with this original strand. Does that make sense? It has to match with this original strand. So you can count your nitrogen bases from the top, right, in the original strand. So this becomes your 1. Actually, this becomes 1. Let me just do this. Do this. There's a This is a nucleotide, first one. 1, 2, just making sure. 2, right? 2, 3, 3, right? Okay. 4, you counting nucleotides. 5, right? 5, right? So this nucleotide 5, or this nitrogenous base A, which stands for adenine, pairs with uh, the nitrogen base 5. You know that adenine pairs with thymine. So our answer here becomes what? Thymine. Okay. I hope I was able to portray how to find the answer there. So you have to be very careful whenever like you looking at such things, right? Okay. Okay, let's continue. Let's continue. Okay. So we've answered 1.4.2b. Let's go to 1.4.3. During which phase of the cell cycle does this process take place? Cell cycle, remember? Cell cycle, during which phase? So the phase becomes what? Interphase, before meiosis, right? So the cell cycle, these are the cycles where the cell is preparing for meiosis or mitosis, right? It's preparing for meiosis or mitosis. So it happens during the interphase. So let's go to 1.1.4. 1.1.4, where in the cell does this process occur? Obviously, this process takes place where? In the nucleus, right? But if you were not able to identify that the process above is DNA replication, you wouldn't have been able to identify the organelle, right? Or the place where this process happens. So this process, DNA replication happens where? Within the nucleus. Okay, let's continue. Let's move on to other questions. Okay, I hope I can be able to draw here. So let's go to 1.5. So remember, guys, as you're preparing for this, uh, for your final exam, so you need to make sure that you study. You need to make sure that you study. Revise all the topics, right? Make sure that you know them and you understand them, especially genetics, right? Because it can be tricky if you did not practice. Okay, let's go to 1.5. In a certain species of butterfly, one gene controls wing spot color and another controls eye color. So this cross is between two characteristics, right? So the first one is what? Wing spot color. This is the first characteristic. And the second characteristic becomes what? Eye color. Eye color. So that's the second characteristic. So we're crossing two characteristics. Obviously, we know that this cross is a dihybrid cross, not a monohybrid cross because we're not crossing one characteristic, but we're crossing two. We're focusing on two characteristics, right? Okay, the wing spot color can be red spots. So we see that red spots are dominant over gray spots, while eye color can be black or brown. So we see that black is dominant over brown. So you know the rules of genetics. We represent the dominant allele with the uppercase letter and the recessive allele with the lowercase letter. So that's the important thing. So please understand that, okay? And make sure that you understand it. Let's go to the questions. Butterflies that are heterozygous for both genes were crossed. So they already told you that the butterflies rather are heterozygous. Heterozygous means that the genes or the alleles are different, right? So let's start with wing spot color. So wing spot color will look like this, right? So the uppercase R, right, and a lowercase r, because they told us that it's heterozygous for both, right? For both characteristics. Then let's go to eye color. Eye color, we have an uppercase, we have an uppercase E, right, and a lowercase e. So you're able to draw this, okay? Okay, so this is heterozygous for both characteristics. So they said heterozygous, hetero means different, homo means same. Okay, so let's go to 1.5.1. Name the type of cross represented above. So the type of cross represented above is called what? A dihybrid cross. Why? Because we're crossing two characteristics. We're focusing on two characteristics. So if they said substantiate your answer or give a reason why you're saying a dihybrid cross, di means two, means we're focusing on two characteristics. Okay, let's go to 1.5.2. Give the dominant characteristic of each gene. So let's start with wing spot color. So wing spot color, red spots are dominant, right? Okay. Now let's go to the second gene, which is eye color. So for eye color, the dominant characteristic, right, is the black, right? It's the black characteristic. Does that make sense? The black color. Let me just say that. Okay. Let's go to 1.5.3. 1.5.3. Okay. 1.5.3 reads as follows. The table below shows the phenotypes and all the possible genotypes of the offsprings. Okay? Possible genotypes of the offsprings. Okay. So now let's go to X. Let's just identify X, right? Okay. So red spots, brown eyes. So let's go back to the statement. Red spots, remember, we know that red spots are dominant over gray spots, right? And black eyes are dominant over brown eyes. So what are possible genotypes of red spot or an offspring that has a phenotype of red spots and brown eyes? So let's start with the first genotype. So the first one will be dominant or homozygous dominant. Let me just write it. Let me write this correctly. So the first will be homozygous dominant for wing spots. Another R here. Okay. Hope you can see that R homozygous dominant. Then let's go to brown eyes. Remember, brown eyes can only be expressed in the phenotype in the homozygous recessive form. So it means you have lowercase e's, both lowercase e's. So two lowercase e's. Because brown eyes are recessive over black eyes. So they can only be expressed in the phenotype in the recessive form, right? So let's try to draw the lowercase e. I'm butchering this drawing. It's hard to draw with this thing, guys, because I'm not using a mouse. Don't worry. I'm sure you're questioning yourself, why am I drawing to this low? Because I'm not using a mouse, and it's very hard to draw without a mouse. But I'll fix that problem just in J. In the coming days, I'll fix the problem because I want to draw it. It's important to demonstrate when it comes to genetics. Okay, yeah, I tried there. Okay, so it's two uppercase R's, because remember red spots are dominant over. Let me check the other characteristic. Let me check it. Red spots are dominant over gray spots. Yes. Okay, let's go to another possible genotype. So I'll just put a slash here. So the other possible genotype is heterozygous dominant. Heterozygous dominant. Heterozygous dominant is just the uppercase R and the lowercase r, right? And still E, still lowercase e, because you can only represent brown eyes with lowercase e. They can only be shown in the phenotype if they are recessive, they're in a homozygous recessive condition. Okay, like this. Okay, okay, let's continue. So already gave you an answer for A, right? So already gave you an answer for A. Okay, let's continue then. Let's go to let's go to B, the phenotype at Y. So the phenotype at Y is red spots and black eyes. So phenotype at Y is red spots. Remember, because the uppercase R, the dominant R is always shown in the phenotype or the physical appearance. So it's the one that will mask the recessive one, right? With regards to black eyes are dominant over what eyes? Let me just check. I keep forgetting these characteristics. Let me go back. That's why it's important for you to always go back to the statement. So over brown eyes, yes. So Y, the phenotype at Y is red spots and black eyes. Okay. Give the genotype of the gametes produced by a butterfly with gray spots and brown eyes. So you can only produce these because okay, let's give the genotype that is produced by butterfly with gray spots and brown eyes. So you can only have this genotype, this one, lowercase e's, right? Okay, let me just start with R. Let me start with R, right? So lowercase r, right? Lowercase r. Because why am I saying lowercase r? Because gray can only be expressed in the phenotype in the homozygous recessive form, and lowercase e. But this is not the answer, guys. I'm just showing you the genotype of a butterfly with gray spots and brown eyes. And this is not the answer because they're looking for gametes, not the genotype. But they said the genotype of the gametes. So this, right, this butterfly can only produce one, right? Can only produce one gamete, right? Or the or similar gametes will produce similar gametes. So it will be E, RE all the way, RE all the way. So I'm sure now you know how to. I'm sure now you know how to get the gametes, find the gametes, right? Because it will take me a long time to show you how to cross the gametes. But you use a monohybrid cross in order to get your gametes, right? So RE, even if you write just RE, right? You just repeat RE 4 times. Does that make sense? So it can only produce this type of gamete. So RE. Hence they're saying here 1 mark, right? Because there's no other option. Okay. Okay, let's continue then. Continue. Let's go to question 2. So now question 2, these are high order questions. These are high order questions, so you need to be alert. Let's go to question 2. Section B, question 2. Okay, the diagram below represents a certain stage of protein synthesis. The sequence of bases is read from left to right. So from left to right, guys, please make sure that you understand that. I'm saying the sequence of bases is read from left to right, right? So it means how would you read uh, this triplet? So it's AGA. So you start from A on your left hand side, you go to your right hand side. So you read it as AGA. So the sequence of bases is read from left to right. So that's what that means. Okay. So a certain stage. Let's look at the stage. What's this stage? So this stage, according to what is displayed here, we can see that this stage is what is transcription. How do we see that it is transcription? Because only one strand acts as a template. So you can see this strand of the DNA, right? It acts as a template. Only one strand, right, acts as a DNA. And also, we can see that the complementary strand that's formed is the RNA. So how do you see that this complementary strand is the RNA? We can see by uracil because we know that we only find uracil in RNA. We don't find it in DNA, right? So you can see uracil here. Okay, so let's continue. Let's continue. Let's continue. So this is transcription, right? Let's go to the question. Identify molecule Y. So molecule Y becomes mRNA or messenger RNA. Okay. 2.1.2. Describe the process that resulted in the formation of Y. Okay, so they want us to describe transcription. Okay, so please listen to me clearly, guys. Make sure that you go revise this process, right? And listen to it over and over again. So the first point is that DNA double helix, don't forget to say DNA double helix. DNA double helix unwinds, right? Then DNA double helix unzips, right? Or DNA unzips, right? And weak hydrogen bonds, they break. After the weak hydrogen bonds break, one strand. Please don't forget that. Don't confuse transcription and DNA replication. In DNA replication, it says each strand, but when it comes to DNA transcription or transcription, let me not say DNA transcription, it says one strand. So one strand acts as a template, right, to form mRNA, right, using free mRNA nucleotides in the nucleoplasm. You can also include the movement of the mRNA. Then after mRNA is formed, it moves out of the nucleus through the nucleopore, right, into the cytoplasm where it attaches to the ribosome. So you can repeat this video over and over again. So it's easy. Transcription is easy. Okay, let's continue. Give one structural difference between molecule X and molecule Y. So let's look at molecule X, right? So molecule X is our DNA because it's double-stranded. I'm already giving you the answer. Then molecule Y is our mRNA, so it's single-stranded. So that's the structural difference, right? So molecule X is double-stranded, whereas molecule Y is single-stranded. You can see here visual that that's the difference. Okay, let's go to 2.1.4. Give the sequence of bases at triplet Z on strand 2. Guys, whenever they're speaking about a triplet, they're speaking about three bases. So now they're saying give the sequence of bases at triplet Z on strand 2, right? On strand 2, okay, so there is strand 2. Okay, so we're moving from mRNA to DNA. So we're starting from left to right. They told us that the sequence starts from left to right. So let's start with U. So U here on Z is going to pair with what? It's going to pair with A. U is going to pair with A, right? Okay. Then A is going to pair with T. Remember, this is DNA. It's a DNA strand. So we're allowed to put T there. But if it was tRNA, we wouldn't, we would just put U there, right? Not T, right? Then C is going to pair with what? G. G, right? So our triplet becomes ATG. So triplet is what? ATG. Our triplet is ATG. Okay, let's continue. That's our answer there. ATG. Okay, let's go to 2.1.5. So this is the question that a lot of people struggle with, but we're going to demystify it today and solve it and try to make it easy. Okay, 2.1.5. The table below shows some amino acids coded for by codons on molecule Y. So they're saying codons. The moment they speak about codons, they're speaking about mRNA. mRNA. So mRNA, right? As you have to be patient with me, RNA. This thing is also writing slow. And next time I'll use a whiteboard, right? After I've fixed the problem of the mouse, I'll just use a whiteboard. So it'll be easy for me to draw, right? But it's mRNA. You get the idea, right? Okay, so let's go to the question. Identify the first and last amino acids coded for by the section of molecule 1. I'll repeat the question again. Identify the first and last amino acid coded for by the section of molecule Y. Okay, let's go to the first. go to the first codon. So the first codon is AGA, right? By molecule Y, remember molecule Y is mRNA. So our first codon or our first triplet of bases, this triplet. Let me just do this. Let me remove this so that you can see clearly. So let's start with our first triplet. So whenever, or codon. Codon is a triplet of bases in the mRNA, remember. So let's start with this triplet. So this triplet is AGA. Now let's look at the amino acid that codes for this triplet. That's coded by this triplet rather. Okay, AGA. Where am I going now? It's moving fast. Hey, hey, yeah, yeah. Okay, sorry guys, be patient with me. Apologies about that. So AGA. So the first triplet is AGA. Now let's look at the table. Which amino acid does it code for? AGA codes for what? Arginine, right? So it codes for arginine, right? Now let's go to the second codon, UAC. This is the second codon, UAC, second codon, UAC. Let's look at our table, which amino acid it codes for. Which amino acid it codes for. UAC codes for tyrosine. Codes for tyrosine. So we found our second amino acid. Let's look for our last codon. So our last codon is CCA. Our last codon is CCA. So CCA codes for proline. So we're done. I think we've answered the question. Identify the first and last amino acids coded for by this section. So the first is, so if we now answer this question, the first is arginine, HEA, and the last is what? CCA, proline. So it's arginine and proline, right? I just gave you an extra amino acid as a bonus, right? So CCA codes for what? The last codon, or the last triplet of bases, codes for proline, right? So I hope I was clear. So let's go to 2.2. The diagram below represents the phase of meiosis 2. So let's identify the phase of meiosis 2. So they're already telling you, they're spoon feeding you, they're already telling you that this is a phase in meiosis 2. So it's easy. Sometimes they don't tell you. So which phase? So we see that our spindle fibers are contracting, right? And they are pulling chromatids to opposite poles. They are pulling chromatids to opposite poles. And which phase does this happen? Obviously anaphase. So it's anaphase what? 2. So this is anaphase 2, right? So we see these structures called what? Centrioles. And this structure, the threads that are coming out of the centrioles are called spindle fibers or spindle threads, either or, right? And also you can see that this is meiosis 2 because crossing over has already happened. You can see that uh, the chromosomes or chromatids are not the same. They're dissimilar, right? Okay, now let's go to the questions. Let's go to the questions. So the question reads as follows. Identify the phase of meiosis shown. So the phase of meiosis shown here is what is anaphase 2. We've already identified it. Anaphase. How do we see? Because chromatids are pulled apart to opposite poles. So let's go to 2.2.2. State 1. Difference between the phase shown in the diagram above and the same phase in meiosis 1. So they're asking us that what's the difference between anaphase 1 and anaphase 2. So anaphase 1, chromosomes are pulled apart, but when it comes to anaphase 2, chromatids are pulled apart. So in anaphase 1, we're dealing with chromosomes. In anaphase 2, we're dealing with chromatids. I hope that's clear. Okay. Let's identify structure A. Let's identify structure A. We're in 2.2.3. Structure A is the centriole. The centriole. Okay, identify structure B. So structure B, these are spindle fibers or spindle thread. Spindle thread. So either or is fine. You'll get a mark. So 2.2.4, describe the role of structure B during cell division. Describe the role of structure B during cell division. Okay, so structure B does what? So it holds the chromosomes or the chromatids in place, right? It holds them together, right? And also it pulls them apart during anaphase 1 or 2, right? So during anaphase 1, it pulls chromosomes apart. During anaphase 2, it pulls chromatids apart, right? So it holds them in place, and also it contracts during these phases, during anaphase especially, contracts, pulling chromatids or chromosomes apart, right? But the most important function is that it holds chromosomes or chromatids in place, right? Whether it might be during metaphase 1, metaphase 2, and in anaphase now it changes its function. Now it pulls the chromosomes apart. Does that make sense? Okay. Okay, now let's go to 2.2.5. Draw a labeled diagram representing the cell above as it would have appeared in metaphase 2. So in metaphase 2, let's try to draw that. Metaphase 2, remember, metaphase 2, you have your, so this is our central, right? Let's draw centrioles here. This is big, okay? Then let's draw spindle fibers, okay? Spindle fibers, okay? Spindle fibers. Then let's draw our chromosomes. We just extend these spindle fibers. Remember, you're not supposed to have space. Let me choose. Okay, if I can draw a nice chromosome here. Okay, so first chromosome held by spindle fibers. I tried to draw it there. Remember, also you should draw the patches of, because the chromatids are not the same, because crossing over has already happened, right? Okay, okay, it's working out. Okay, so this is how the face would have appeared. Let's just link it. Yeah, okay. So our face would have appeared here. Now let's look at the color. Don't forget to color these chromosomes, guys, because they're not the same. They're not the same. So where will you get the color? You'll get it from the face that's shown above in the diagram. So we can see here this one. We can see that this one is patches of white and black, and this one is patches of. So this one is more black than white. This one is more white than black. So these ones are just black and white. So let's try to do that then. So we can see that this one is colored. Okay, guys, it's going to be messy when I color. But where do you get the color? You get it in the chromatids that are shown where in the face there. But you get the drift, you get the idea, right? So this is how you would draw your stage, right? Okay, so don't forget to do that. Okay, okay, let's just leave it like this and continue. So I don't want to prolong this video as it already is long, right? I want to prolong this video. Okay, let's go to 2.3. The table below shows the percentage of blood donors for each of the blood groups in South Africa in 2018. Okay, so let's go to the question because the table is easy, right? They've already analyzed everything for us. So they gave us the blood group and the number of donors, right? So we have blood group O with 48 donors, A with 38 donors, B with 10 donors, then AB with 4 donors, right? Okay, so 2.3.1. According to the data, which is the most common blood group amongst the donors? So the most common blood group is blood group O, right? So that's the answer. 2.3.2. Name the type of dominance shown by the inheritance of blood group A. So inheritance of blood group A shows a complete dominance, right? Complete dominance. So blood group A, B, they show complete dominance, right? But blood group AB, right, they show codominance. So yes, why blood group A? Why does blood group A show complete dominance? Because it masks, right, or it hides the blood group O allele, right, the recessive allele. In that state, blood group A will dominate over the O allele, or the allele that causes blood group O, right? Okay, let's go to 2.3.3. Explain how it is possible for a man with blood group A and a woman with blood group AB to have a child with blood group B. So this is possible, right? So explain how. So if let's go to the man. Let's start with the man, not the female or the woman. Start with the man. So the man will have, uh, let me just scroll down like this. So the man will have, right? The man will have this blood group. I'll just draw it first, then I'll explain it after, right? So the man might have this genotype. Come on. So the man might have this genotype, which is the uppercase A and the recessive allele, right? Men can have this genotype, or he can have this genotype, this one, right? The homozygous dominant genotype, or the homozygous dominant, right, genotype. So when the child, right, the child will inherit either of these alleles, right? The child, in this case, the child could receive either the A allele, the dominant A allele, or the recessive O allele, right? Or in this case, the child might receive either the dominant allele, or so the child in this case, if the man has this genotype, this homozygous dominant genotype, he cannot uh, pass on any other allele besides the dominant A allele, right? But when it comes to the woman has only one. Woman has only one genotype. So there's only one genotype for AP. This is the genotype, all right? This is the genotype. AP. Remember AP, they are codominant. They are both expressed in the phenotype equally. So the woman, right? So the woman can have this. They can only have this genotype, right? So let's say we have uh, a blood group A, right? So the male, let's say the male donates, right? The A allele, right? A allele, the A allele, right? The A allele, right? And the woman donates the B allele, right? The B allele, the B allele, the B allele. Right, then the child will be AB. So we let's grab this genotype because it can never happen, right? No matter how we dice it, so it can never happen. So the child will never have a blood group B if the male has this genotype for blood group A. So the male, which leaves us with this genotype. it leaves us with this genotype, this one. Let me remove this one. It leaves us with this genotype. So with this genotype, it's possible for the male and this woman to have a child with blood group B, because the child would inherit this, the child can inherit this recessive allele from the father, right? I'm already explaining, guys. The child can receive this recessive allele from the father, and the mother can donate this allele, or the child can inherit this allele from the mother, this B, this dominant B from the mother, right? Let me just place them correctly. We start with the dominant one. Remember, we're starting with the dominant one, right? So start with B, right? So B. So this is from the mother. The child will get that group B from the mother, or the B allele from the mother, and get the recessive allele, or the O allele. I call it the O allele, right? From the father. Then in this case, both parents, right, can have a child with blood group B. I understand. So both parents can have a child with blood group B, but that can only happen if the father has this genotype, right? This genotype where A is dominant over the recessive O allele, right? Then the child will inherit the recessive I allele from the man or from the father, and receive the B allele from the mother, because the mother only has one genotype, can only have one genotype. So I hope that makes a lot of sense, even though I couldn't display it properly, but I hope it makes a lot of sense, right? So we cannot have the other genotype. The only genotype a man can have, right, is that one, right? That is, I call it the heterozygous A genotype. Okay, so let's go to 2.3.4. So 2.3.4 reads as follows. Plot a bar graph to represent the data in the table, right? Okay, so it's easy, right? So you have your blood groups. I will explain the graph, but let me try to draw it though. Let me try to draw it, even though it will be ugly. So we have our graph here. So this will be our x-axis, right? Or our horizontal axis. And this will be our y-axis or our vertical axis. So here, let me just look at the table again. So here in our x-axis, we'll have blood groups. So in our x-axis, we'll have blood groups. So you have blood group O, right? Blood group A, right? Blood group AB, and blood group B. Okay, so this is just a rough sketch of our uh, bar graph. Okay, then on the y-axis or your vertical axis, you have donors. Your donors in percentage, obviously. Donors, writing some other way here. So you have donors, right? Donors. So what's important here is to have your scale, right? Make sure that your scale has equal intervals, right? So which scale can we use here? I'm removing the whole thing now, but it's fine. Right? So you have your donors on your y-axis and your blood groups on your x-axis, right? You have those. Uh, Next time, guys, I'm using a blackboard and I've sorted this mouse thing, right? Okay, so which scale will we use here? So let's see. So we can use 0, 2, 4, 6, 8, so right. So you can use that 2, right? The 2, 4, 6, 8, 10, 12 table, right? Or scale rather, a table scale. Or you can use your fours, right? 0. Remember, start with the 0. Right? Don't just start with the 4. 0, 4, 8, 12, 16, 20, right? So you can use that, right? So use it until the 2 table, you can use it until 50, right? You can count until 50 because now here the maximum value, let's look at the maximum value. So the maximum value is 48 and the minimum value is 4, right? So it means that you have to end at 50 just to accommodate 48, right? Or just use 4, guys. You can use 4. Just use 4. Just use 4, right? Just use 4. Hope that makes a lot of sense. Just use uh, multiples of 4. So that will be easy because you don't want your graph to be long now, or you don't want your y-axis to be long. Just make sure that you use multiples of 4, right? Okay. And also the important thing is that you should have a caption that will include both axes. So your caption should include your x-axis, which are your blood groups, right? And the percentage of donors, right? Percentage of donor. So it should have that, right? And guys, a bar graph has spaces in between the bars. So there are spaces in between the bars, equal spaces in between the bars. Never forget that. Equal spaces in between the bars. So this is not a histogram. It's a bar graph, right? Okay, let's continue. Okay, let's go to question 4. Why am I saying question 4? 2.4, not question 4. 2.4. 2.4. These questions are long, guys. These questions are long. My brain is a bit uh, sleepy now, but don't worry, guys. Don't worry. We're used to this. Okay, 2.4. Muscular dystrophy is a genetic condition that causes muscles to weaken over time. It is caused by recessive allo. And remember, guys, whenever they say a condition is caused by recessive allo and it's sex linked, right? It means that it affects males more than females, right? Always remember that. So the moment they say it's caused by recessive allo, it affects males more than females. Why? Please write this down. Please write this down. You can repeat this video over and over again. Why? Because females can be carriers, right? So females have two X chromosomes. Let's start with females. Because females have two X chromosomes. So they can be a carrier in a condition of a heterozygous form, right? By heterozygous form I mean this. They can have this, right? So this is heterozygous and you got D here, okay, another X, then a smaller d. Or it could be no more like this. Right? It could have dominant or dominant D, right? Homozygous dominant, right? Like this, right? So females could be carriers, right? Because they have two X chromosomes. Whereas males, they can't be carriers. They have one X chromosome. And this disease affects X chromosomes only. So when you're a male, you're either affected or not affected. Whereas in females, they can be carriers. So it means that in a heterozygous condition, they can still be normal. Right? They can still be normal. When it comes to males, you don't have that. They're either affected or not. So that's why sex-linked disorders or diseases that are caused by recessive allo can affect males more than females. You have your color blindness, your hemophilia, right? Even this one, it affects males more than females. Okay. pedigree diagram. So how do we attack the pedigree diagram? How do we analyze it? How do we analyze the pedigree diagram? Let's look at the key there. Before we analyze, let's look at the key. Let's look at the key there. The key says that when you have a clear square, that's a male without the dystrophy, right? When you have a shaded square, it's a male with muscular dystrophy. When you have an unshaded circle, it's a female without muscular dystrophy. When you have a shaded circle, it's a female with muscular dystrophy. Okay, so one, let's start with one. So the first thing that you do here, you mark all affected individuals. It's easy to mark affected individuals, especially if they're male. They only have one genotype. Remember, when they're male, it's either affected or not, right? So let's mark number four. Let's go to number four. Number four, it will be a smaller h. With h, are they using h? No, they're using d. Now I have a hemophilia in mind. So they're using D. So it's a smaller d and a y. Mark all affected males. So we can start with males, it's fine. The D is supposed to be at the top, but you get the idea, right? So we're done. So let's mark 8. So it means even 8 and 7 will have the same, right? They will have the same genotype. They have the same genotype, right? What's happening? What's happening now? Smaller glitch. Genotype. Small glitch. Fix the problem. More the affected male. And all because they also have one. Now let's go to unaffected male. They also have one. I think I've fixed the problem. No, good. Okay, yeah. And also the unaffected males. So they have one. What is this thing doing now? My laptop though. Okay, guys, let me just do this. You do this. Okay, just fix the problem, guys. Let's fix the problem. I think I'm going to end it here, guys. Or before I end it, right? Let me just go to evolution because that's the problem most people they struggle with, right? You just go to evolution before my laptop switches off, right? Let me go to evolution because I see this um this laptop is going to cause problems, and also my battery is about to be low, right? So let me just go to evolution 3.4. Let's go to 3.4, right? Because I know that most students they struggle with evolution. Okay, let's go to 3.4. The passage below describes Homo erectus, but does it start here? 3.4. Yeah, it starts here in 3.4. Okay, let's go to 3.4.1. Describe how the fossils of Homo, right, erectus are used to support the out of Africa hypothesis. So, what is the out of Africa hypothesis? The out of Africa hypothesis states that all organisms, right, all organisms or Homo organisms, right, they all came from Africa, right? All organisms, they came from Africa and they migrated to other parts of the world, right? All human ancestors, all human ancestors, they they started in Africa, right? This is where they were born in Africa. They were born in Africa and they migrated to other parts of the world in later stages. So they're saying here, describe how the fossils of Homo erectus are used to support the out of Africa hypothesis, right? Okay, so the oldest fossils of Homo erectus were found in Africa. The oldest fossils of Homo erectus were found in Africa. Then the uh, youngest fossils of Homo erectus were found in other parts of the world, like Asia, which means that they migrated to other parts of the world, right? So the oldest Homo erectus fossils were found in Africa, and the youngest were found in what? In Asia, right? In other parts of the world, right? Where they migrated means that they migrated later at a later stage to other parts of the world. Okay, let's go to 3.4.2. Describe how other fossils evidence is used to support the out of Africa hypothesis. So how do you attack this question, guys? How do you attack it? So the first thing that you say, you start with Adipithecus fossils, right? Adipithecus fossils, all those fossils they were found, they're only found in Africa, right? Then we move from Adipithecus, we go to Australopithecus fossils. They were only found in Africa. Then we go to a Homo habilis. Oldest fossils of Homo habilis were found in Africa, or Homo habilis, right? Then we go to Homo erectus. Okay, we're no longer including Homo erectus because we've already spoke about Homo erectus. Let's go to Homo sapiens. The oldest fossils of Homo sapiens were only found where? In Africa. Does that make sense? And the youngest fossils of Homo sapiens were found in other parts of the world. That's how you answer such a question. Okay. So let's go to 3.4.3. Use the information in the passage regarding Homo erectus and tabulate differences between the features of Homo erectus and Homo sapiens, right? Okay, so let's start with the first one, right? So Homo erectus, they were starting to erect, hence they're called Homo erectus, which means that they were still they did not have an upright posture, right? So Homo erectus did not have an upright posture, whereas Homo sapiens had an upright posture. Does that make sense? I hope it makes a lot of sense. Then Homo erectus, right, had a smaller cranium. Remember, Homo sapiens are called the wise men. So Homo sapiens, sapiens means the wise man, right? So Homo erectus, they had a small cranium, whereas Homo sapiens had a larger cranium, just to show the difference in intelligence and wisdom. Does that make sense? Right? So two differences. So I've tabulated two differences. Does that make sense? So the first difference is that they did not have an upright posture, right? Whereas Homo sapiens had an upright posture, they were able to stand up straight. Another difference is that Homo erectus had a small cranium, Homo sapiens had a large cranium. Does that make sense? Does that make a lot of sense? I hope it makes a lot of sense. You can also include the effect of brow ridges, right? So they had protruding brow ridges, right? Homo erectus had protruding brow ridges, whereas Homo sapiens did not have protruding brow ridges, right? So our brow ridges are flat, right? They're not protruding, they're flat. Whereas Homo erectus, their brow ridges are protruding, or they are prominent. Let me just say prominent. Okay? 3.4.4. What observation of the skull of Homo erectus would have led scientists to conclude that Homo erectus was bipedal? So definitely, so this one is easy. So it means that the position of the foramen magnum, right? Foramen magnum, the position of the foramen magnum in the skull was in a forward position. Well, if it was in a backward position, then it will mean that they were quadrupedal. They were not bipedal. They were not able to stand up straight. They were not able to walk on 2 feet, right? But for the fact that the foramen magnum was at a forward position, it means that even their spine was as shaped like the one of Homo sapiens. Okay. Okay, let's go to 3.5. 3.5.1. What is a mutation? Remember, what is a mutation? It's the change in the sequence of the gene. Or you can say that it's the change in the nitrogenous basis of the gene. Okay? Okay, let's read the statement because now I'm rushing. Let's read the statement so that you don't get confused. In 1986, there was a huge radioactive disaster in Chernobyl, and all humans immediately left the area because of the deadly high radiation. Animals exposed to high radiation developed cancer or died. Almost 40 years later, scientists have been studying the wolves that live in this highly radioactive environment where they are exposed to high radiation. They found that those wolves have a mutation that makes them immune to cancer. Okay, okay, so that's the paragraph there. That's the extract. So let's go to 3.5.2. What effect does exposure to high radiation have on animals? So high exposure to radiation causes cancer, right? So it causes cancer. Okay. 3.5.3. Use Darwin's theory of natural selection to explain the development of immunity to cancer in these wolves. Okay, now they want us to use natural selection, right? So natural selection, we're starting with variation. So variation, there was a range of different wolves within the species of wolves, right? Within the population of wolves. Does that make sense? There was variation. So what's that variation? It means that there were wolves who were resistant to cancer and there were wolves who were not resistant to cancer. Does that make sense? And when the wolves were exposed to high radiation, as it is said there in the article, when they were exposed to high radiation, those who who were resistant, right, were resistant to cancer, were able to survive the radiation and they did not develop cancer. Whereas those who were not resistant to cancer, they developed cancer, right? Does that make sense? So those who developed cancer, they eventually died because cancer kills. Does that make sense? So they eventually died. And those who survived, remember, natural selection is called the survival of the fittest. And those who survived, they reproduced and passed on the gene of resistance to the next generation. Up to the point that in the next generation, you only had the wolves that were resistant to cancer. So that's natural selection. You can repeat this statement over and over again, or repeat this video over and over again. But I just wanted to touch on evolution before, right? Because my laptop was just glitching, right? My laptop was glitching, right? But let me look for another question I can do. Let me look at the questions. Experiments, yes. Let me do an experiment. Okay, because a lot of people struggle with experiment. 3.3. A group of researchers conducted an investigation to determine the influence of the type of milk, right? The influence of the type of milk. So already the type of milk becomes your independent variable on the height of the children. The height becomes what? What's being measured? The height. The height becomes the dependent variable. Already they're telling you. So the statement will always tell you the independent variable and the dependent variable. Remember, always remember that. The investigation was conducted over a period of 7 years from age 3 to age 10. So that's reliability. Remember, reliability is about repeatability and sample size. Okay, let's continue. They selected 4,146 healthy 3-year-old children of the same race and gender. So the sample size of 4,146 is uh, reliability, right? So they are making sure that the investigation is reliable. And also the same race and same gender, it's validity. Whenever they say same, that's validity. Okay? Divided the children into two equal groups. One group was given 1 liter of cow's milk to drink daily. The other group was given 1 liter of soy milk to drink daily. Ensured that each group had the same diet, same additional diet, similar daily activities, similar living conditions. They are ensuring validity when they are doing these things. Remember, whenever they say same, similar, they are ensuring validity because if they don't do that, it will compromise the validity of the investigation. Okay, let's go to the next one. Provided the milk from the same supplier, validity again. measured the children's height at the end of the investigation and calculated the average. Okay, so let's look at the table. So it says that expected average height of the child 146,86. Average height of the child on cow's milk 141,4, and average height of the child on soy milk 147,4, right? So we can see that the cow's milk, right, has more calcium. We can say that it has more calcium than the soy milk, right? Because now the child is growing taller, right? It's growing taller when they drink the cow's milk than as compared to when they're drinking the soy milk. So already I gave you the conclusion of this investigation. Okay, we've already identified the independent variable and the dependent variable. So the independent variable is the type of milk, the type of milk, and the dependent variable is the height, right? It's height. Okay, 3.3.2. Name the type of variation displayed by height of a person. So we have two types of variations. We have discontinuous variation and continuous variation. Continuous variation, we have intermediates, we have in-betweens, right? And height has in-betweens. We can have a height of 4,5, 6,5, 62,5. But when it comes to discontinuous variation, you have categories, right? You have distinct differences, like blood groups. It's either you have blood group A, B, O, or AB. So you can't have an in-between. So that's the difference. So here, the type of variation is what? A continuous variation. Okay, state 2, 3.3.3, state 2 planning steps that the researchers had to consider at the beginning of this investigation. So 2, obviously the venue. They had to determine the venue. They had to ask for permission. They had to ask for permission, right? From their parents, from these 3-year-old parents, right? So they had to ask for permission, right? They had to look at their medical records before they conducted this investigation. Does that make sense? Okay, also the equipment that they were going to use. So they had to pay close attention to the equipment or buy the equipment that they were going to use as they were planning. Okay, 3.3.4. State 3 ways in which the validity of this investigation was state 1 factor. State 3 ways in which the validity of this investigation was ensured. Okay, let's look for similar, same. Okay, the same diet, they're giving the same diet, right? Similar daily activities and also similar living conditions. Also, you can add there, it was the same race and same gender, right? So same race and same gender. Okay, there are a lot of things you can say here. It's more than 3. So 3.3.5. State 1 factor that could have negatively affected the validity of the investigation. If they gave them a different diet, it would have invalidated the whole investigation, right? So if they had different living conditions, right? If they did not get the milk from the same supplier, then it would have invalidated the whole investigation, right? So there are a couple of things you can say about this. How did the researchers ensure reliability? Remember, sample size. They used, don't ever say sample size. Whenever you're answering this question, just say that they selected 4,146 healthy 3-year-old children. So that's how they ensured reliability. So, okay, state the conclusion of this investigation. So the conclusion is that when a child is on cow's milk, they grow more than the child who's on soy milk, right? So, or you can say that the cow's milk contains more calcium, right? More than the soy milk. So that's the conclusion of the investigation. So I'll end it here, guys, and I'll stop the video, right? I hope this was helpful to you. And thank you so much. I couldn't continue with this because now my laptop is glitching and also my battery is about to just degenerate, right? It's about to switch off. So thank you guys. Let me stop this video. I'll publish it and you can repeat it over and over again. Doesn't matter how many times, as long as it helps you. Thank you so much. And I hope we can do more of this with regards to um, drawing, including more drawing and everything. I'll try to fix that issue, right? And try to write on a whiteboard after I fix the What's this? The mouse issue, right? Thank you, guys.